Good afternoon. We will begin. So we're very fortunate to have a guest today, Dr. Jonathan Wolpaw, who has come to us from Albany, New York, where he is the director of the National Center for Adaptive Neurotechnologies and chief of the Laboratory of Neural Injury and Repair at the Wadsworth Center, which is part of the New York State Department of Health and linked to the State University of New York in Albany. Dr. Wolpaw received his MD degree from Case Western Reserve and then took training in medicine and neurology. He's a certified neurologist and also in investigative neuroscience, which included time spent here and also across the street at the Armed Services. He's a leader in one of the more, I think, exciting, imaginative frontiers of medicine today, which closely links bioengineering, computers, neuroscience, and patients with a variety of neurologic diseases. In thinking about just saying a couple of words in introducing Dr. Wolpaw, I was struck by yesterday the fact we had Siddhartha Mukherjee, the author of the Pulitzer Prize-winning book, Cancer, the Emperor of All Illnesses, and the more recent one, which I'd encourage you all to read, called The Gene. And the point of bringing up this reference is that with the development of technologies, skills, knowledge, in areas that become vital to medicine, issues arise which spill over into the arena of ethics, of adaptation. In the case of genomics, it becomes the whole question of selection of people in the old days who were to be exterminated, or in modern times modifying genes for various cognitive functions. And to a limited extent, we haven't gotten that far, but the techniques of brain-computer interface raise or will raise some of those questions, which will emerge, I hope, in our discussion. Dr. Wolpaw's work has been in the forefront of studies of modulating spinal cord plasticity in experimental models and in humans to produce functional outcomes to be desired, like someone walking, for example, or walking better. And more recently, in the past about 25 years, with the birth of this enormous interest in brain and computer interfaces, he and his colleagues have demonstrated the usefulness of these techniques in experimental models, and we'll hear more about where things stand. The reference to the societal aspects, the ethical aspects, are not just totally out of air, because he just told me that very shortly he will be going to a meeting, I think, in New York, directed entirely to the question of what are the limitations or what rules may or should be set up for the interfacing of the computer with chips in a brain or functioning of any neurological system. This is sort of analogous to the problems at the time of the Asilomar Conference for Recombinant DNA and now with the big issues arising about gene editing. So we're very grateful to you for traveling the long distance from Albany, and we look forward to your talk. Thank you, John.
So thanks, Win. So I'm very happy to be here. I want to thank Dr. Arias and Dr. Pettigrew for inviting me to come down here. It's great to be at NIH, which we are largely beholden to for our research support for the last 35 years. At present, we're in the third year. Let's see if this, oh. Okay. The National Center for Adaptive Neurotechnologies is a P41 biotechnology resource center that's funded by NIBIB. We're in our third year. And we've also had very generous and stable support for over the last 35 years from NINDS and also from NCMRR, the National Center for Medical Rehabilitation Research of the Institute of Child Health. So most of what you'll see, the work that we've accomplished, what we're engaged in now, has been supported by NIH and also more recently by the Veterans Administration. So we're, as Wynn said, we're at the Wadsworth Center, which is the laboratory arm of the New York State Department of Health. New York State is unusual in having a very large laboratory facility that does both regular clinical services for doctors and hospitals throughout the state, as well as a large research component that's been there for more than 100 years now. The drug nystatin that I think many of you are familiar with, the name stands for New York State. It was discovered at the Wadsworth Center about 50 years ago now. In any case, I'll be talking about what we now call adaptive neurotechnologies, rhythms, reflexes, and rehabilitation. And this, what I'm talking about, has really been made possible by two relatively recent advances. And by recent, I mean in the last 30 to 40 years. So the first of these advances is the recognition that the central nervous system changes throughout life, that activity-dependent plasticity is occurring everywhere. So for people who weren't in research or in neuroscience 30 or 40 years ago, it's really hard to appreciate how remarkable the change has been over that time. Back then, we all knew the nervous system was pretty hardwired. It changed very little and only in very selected sites. Therefore, if you had damage, you had disease, you were pretty much stuck with what was left. And you had to work around what was left. There was really not much ability to make actual substantive changes, to rebuild, to regenerate, to regain what had been lost. So that's all been changed since we now understand how plastic and continuously plastic the central nervous system is. So the other advance is hardware and software. Yes. Okay. Hardware and software that enables us to have complex real-time interactions with the central nervous system to initiate and guide this plasticity. Now again, this hardware and software either didn't exist or it was enormously cumbersome and expensive even 20 years ago. I mean, I remember when I was first at NIH in the 1970s, there were still around some of the old adding machines, the one with the wheels that whirl and all the numbers come up. It's all very satisfying. You hear this clicking and a lot of sound, and then your answer comes up. They weren't actually being used still, but the mechanical machines were still around. That's changed, and now we have hardware and software that allows us to have very complex real-time interactions with the central nervous system easily, quickly, inexpensively. So these two advances together have given us unprecedented opportunities for establishing new kinds of adaptive interactions with the nervous system, that we can gain new insights, new basic science insights, and we can also develop new therapies. And what I'll be talking to you about is very much the two are intertwined. Most of the work that we'll be talking about really started as basic science, and to a considerable extent it continues as basic science, but at the same time, in the last decade or so, it's developed into actual applications. And as it turns out, when you actually develop into clinical therapies, and you're still engaged in the basic science, the two areas really inform each other and they reinforce each other. So, adaptive neurotechnologies. As far as we know, the purpose of the CNS, really the sole purpose of it, is to interact with the outside world in the body through peripheral nerves and sensory receptors and muscles. That's what, as far as we know, it does. Adaptive neurotechnologies replace, restore, enhance, supplement, or improve these natural interactions. They typically adapt their behavior to the particular CNS that they're interacting with, and they often induce adaptive plasticity in the CNS. So I'm going to be talking about 4 different kinds of adaptive neurotechnologies, primarily the kinds that we've been very much involved in. They overlap to some extent. Some of the principles overlap, but they're very distinctive, primarily also in terms of the applications to which they're put at present. The first are brain-computer interfaces for restoring communication and control. The second are brain-computer interfaces for rehabilitation, specifically in this case for stroke rehabilitation. The third area is using electrocorticography, which is the recording of signals actually from the surface of the brain for pre-surgical mapping of cortical function. And the fourth is reflex conditioning for rehabilitation after spinal cord injury. And this introduces really a new therapeutic class for rehabilitation, which we refer to as targeted neuroplasticity. And I'll explain more what I mean by all these as we go along. So first, what is a brain-computer interface? As I just set up above, as far as we understand it, the CNS produces outputs that act on the world or the body. All the natural outputs are neuromuscular, or they consist of the excretion of hormones. Those are the natural ways the CNS acts on the world, the internal world and the external world. A BCI measures CNS activity and converts it into artificial output that replaces, restores, enhances, supplements, or improves natural output, and thereby changes the ongoing interactions between the CNS and its environment. So this is a diagram of a brain-computer interface and the things it can do. So here you see record it. You can record brain signals from the surface with EEG, for example, from the surface of the scalp, from the surface of the brain with ECoG, or from within the brain with microelectrodes that record single neurons or local field potentials. So you acquire these signals, and then they're analyzed to extract various features. So these features can be the firing rate of individual neurons, they can be the latency of a particular evoked potential, they can be the amplitude of a particular frequency band at a particular location. These are features that are extracted from the brain activity, and then they get translated into commands, which produce outputs that control really these 5 kinds of applications. And these 5 categories really cover anything that you can imagine a brain-computer interface doing. The 2 that have received the most attention in the last several decades are BCIs that can replace loss function, for example, to operate a wheelchair for someone who's paralyzed, or to operate a prosthetic arm for someone who has had a spinal cord injury, or BCIs that can restore function. They use brain signals to stimulate the nerves or muscles to restore control to a paralyzed arm. But there are 3 other potential classes of BCIs. One are BCIs that enhance normal function. So if you have someone who's supposed to pay attention continuously, and you look at his brain signals and you can tell when he's not paying attention, or you can predict when he's about to not pay attention, you can make sure he doesn't lose attention. So you can enhance normal function potentially with a brain-computer interface. You could, in theory, supplement function. Here you see a third arm, a robotic arm, and you could use brain signals to provide independent control of a third arm. And finally, you could potentially use brain signals to assist in rehabilitation, to guide plasticity, to enhance, to augment the efficacy of rehabilitation methods. And we'll get back to that a little later. But the most attention in the past and actually at present is still on the replace and restore categories. And this shows a very simple kind of BCI for communication. to restore communication to people with advanced ALS, for example, or high-level spinal cord injury, people who are almost locked into their bodies and really can't communicate in any substantive way. So you see here a matrix of letters and numbers and function calls. There's 72 items. It's a fully functioning keyboard. And here you see, this is an email application, an email that's being composed. So far the person has spelled H-E-L, and this is a predictive speller to allow the person to go faster. For example, by selecting the 1, the hello can be completed faster than having to spell the L and the O. So what you'll see is that groups of these items flash very rapidly. And the BCI looks at the EEG responses to these flashes. And it can tell from those responses which item the person is paying attention to, and therefore which item the person wants to select. So it doesn't require that the person look at the item, so that it can conceivably function in the absence of any eye movement. It requires that the person pay attention to the item. And when that happens, the response is different, and the BCI, after a series of trials, can recognize that difference. So here you'll see it in operation. So there's the flashing. And he selected the 1, so now you see hello. And he selected W. This is in real time. So you see this is kind of slow, but it functions. And if you didn't have any other means of communication, it could be very useful. So he selected the number to give him hello world. And now he selects the exclamation point. So for example, if you're fairly good at this, you can do this fairly effectively, and it varies from person to person. You could certainly write 100 words in an hour, which is slow. But in thinking about that, I realized that when I write, in any case, papers or grants or whatever, I write about 100 words an hour. So it actually wouldn't slow me down to have to use this to write. So that's a very simple BCI, and this shows how it operates. This is a recording EEG through a laptop to the user screen. This is what the computer is looking at. This is the response after the flash, and you see when the flashing group includes the item the person wants, you see this big response. It's called a P300 or an AHA response. It was originally used in this way for communication by Manny Donchin about 30 years ago now. And the green is what happens when items flash that the person doesn't want. So after a series of trials, the computer can very definitely, from the 72 item matrix, figure out what item the person wants to select. This is the top of the head. It shows the distribution of the difference here. And it tends to be focused posteriorly and centrally over the head. So this is where the discrimination is. This is the way this P300 BCI works. And it's very simple, but it functions. And we've just completed actually a study, a VA-sponsored study, looking at its use by people with advanced ALS in their homes. So people used it independently with their caregivers in their homes. We had a variety of applications. This is a YouTube application. Again, you see the same matrix, but with a different application. We have training videos. This is for training caregivers how to put on the cap. This is one of the people we work with very closely who has ALS. We've worked with her for many years. This is her caregiver learning how to put on the recording cap. And this is a message that was sent to me by an email that was sent with the BCI to me by actually our very first person who we gave the BCI to in 2006. This was a NIH-funded researcher who had advanced ALS and was unable to use his eye movement system anymore. And he used the BCI for a number of years to communicate with his children, to write grants, etc. I asked him the impact of the BCI on his life. He wrote back, "No problem. I couldn't run my lab without BCI. I do molecular neuroscience research, and my grant pays 3 people. So this was a very succinct but eloquent statement of the impact on his life. Yes?
Very briefly describe what ALS is.
Oh, amyotrophic lateral sclerosis. It's a progressive disease in which motor neurons in the spinal cord die. There can be involvement of the cortex as well, but basically people become gradually more and more paralyzed. It generally has they generally die within several years unless they accept long-term ventilation. There are people such as Stephen Hawking who live for very long periods, but that's very rare. The potential impact of the BCI is that most people, more than 90% of people with ALS in this country and generally throughout the world, they don't elect to accept permanent artificial ventilation, and they die when they can no longer breathe. So that's something over 90%. There are relatively few people who elect to keep living as they become more paralyzed. However, there are a number of studies now indicating that the quality of life for people who are that paralyzed is not necessarily any worse than the quality of life of the rest of us. They can lead enjoyable, even productive lives if they can be well supported, they have a social situation, they can continue to communicate. So communication is a big issue for them. So things like methods like this that could continue communication for people in this state could potentially have an impact on clinical practice. So in any case, that's ALS. So another kind of signal that can be used for communication are sensory motor rhythms in the EEG. Now this is the top of the brain. The nose is at the top. This is looking from the top. You see here is the sensory and motor cortex on both sides of the central sulcus. Now these areas of cortex are most directly concerned with movement and with sensation, and they produce rhythms that are referred to as sensory motor rhythms. So this is one, an 8 to 12 hertz rhythm known as the mu rhythm. They're higher frequency rhythms, beta rhythms, and gamma rhythms. These rhythms tend to be present when the cortex is awake and alert and isn't doing anything. When it starts to be active, when sensation comes in, when movement occurs, the rhythms go away. It's called desynchronization. So these rhythms reflect the functioning of these areas of the brain. And what we and others have shown is that people can learn to use motor imagery without any actual movement or sensation to control the amplitude of these rhythms. And they can use this to control a cursor on a screen. So for example, here's a person who's learned how to make the rhythm bigger to move a cursor up a screen, or smaller to move it down a screen. And the computer simply looks at this, does a frequency analysis, looks at the power or the amplitude in the 8 to 12 hertz range. It's large with a top target, small with a bottom target, and it knows to move the cursor up the screen toward the target. So by mastering this over a series of training sessions, a person can move the cursor up and down the screen at will. And this shows, again, this is the top of the head, the nose here. You can see the focus of control here primarily over right sensory motor cortex. So it's this location that's being used. And in another person, it might be a slightly different location. So the training, this is part of the adaptation. The training period is adaptation of the person as well as the system. The system has to adapt to what the person can best control. The person has to learn how to give the computer what it can best interpret. So there's you want a beneficial a mutual interaction of the two. You have two adaptive controllers that need to adapt to each other in an ultimately beneficial fashion, the brain and the BCI. And that's actually why BCIs are also called brain-machine interfaces. It's a frequently used term, but I prefer computer interface because it definitely includes the concept that the computer is adapting. Machine implies that it's more inflexible, but it's clearly an adaptive system. So we've gone on to show, and this is primarily Dennis McFarlane's work in our group, that people can learn to control different sensory motor rhythms at different locations simultaneously to provide multi-dimensional control. So here's a signal that this person has learned to control to produce vertical movement. At the same time, she's learned to use a signal here to produce horizontal movement and a signal here to produce depth movement. And again, the distribution, the exact rhythms, the exact locations vary from person to person. And here you can see this actually working. This is a virtual three-dimensional cube. The cursor starts in the center of the cube and there are targets at the four corners. And this is in real time, and she's learned to control, and this is independent control. We were able to show that. She can control the 3 dimensions simultaneously and move to the targets. So clearly this works, but at the same time you're going to notice that it's not perfect. It's slow, it's kind of jerky, sometimes she's more successful than other times, and this is a feature of BCIs for any sort of complex control. It's not just a feature of EEG-based BCIs, it's a feature of BCIs that use implanted electrodes, that look at single neurons, that look at local field potentials, that look at ECoG. You can get very nice demonstrations of significant control, but you don't get control that is as rapid and, most important, as reliable and consistent as normal motor function. And that's something that was originally ascribed to what we need better resolution, but even with better resolution in terms of recording, you still have the problem of reliability and consistency. And to me, the reason for that is what I call the Grand Canyon problem. And this faces really all BCIs at this point that are used for communication control. So here you see someone standing at the edge of the Grand Canyon. Now, unless you're a person with a death wish, there's no way that you would ever use a BCI to control your movements at the edge of the Grand Canyon, either in a wheelchair or control your muscles in your body. Unless you were courting death, you wouldn't trust a BCI to maintain your position here at the edge of the Grand Canyon. So until that happens, BCIs to restore or replace normal control are not going to really come out of the laboratory in any very meaningful way. And yes?
What about effort level after somebody's trained?
Yeah.
Is it hard?
Well, not necessarily, no, because initially, as I mentioned, people use motor imagery to do this. But as they get better and as they go into 2 or 3 dimensions, they can't tell you how they do it. They do it like you raise your arm. It becomes more natural, but it doesn't become as reliable as raising your arm. So in any case, I think that this is the primary origin of the Grand Canyon problem. So this is a very, obviously, vastly oversimplified diagram. Yes?
Do people with...
Could you repeat the question?
Oh, yes, okay. The answer to your question is yes, I think. Victoria.
Do people with improved physical aptitude in 3D normal space have improved neuronal targeting aptitude?
No, we don't know enough, or we haven't done enough subjects to know. I thought you were going to ask if people who are paralyzed can do this, and yes, they can. Yeah, at this point, we don't know. I think to some extent it depends very much for EEG on the recording characteristics. If your rhythms are very easy to record from your scalp for some reason, then it's going to be easier for us to do this and easier for you to do it. Yes?
How do you train people?
We give them, we start with a session in which we ask them to move move one hand, move the other, move their feet. We see where their sensory motor rhythms are located and what amplitude ranges and what frequency ranges they are. There are relatively reproducible, people are very similar, but they're not identical. So we'll say, okay, this person has a lot of desynchronization at 8 to 12 hertz at location C3 or CP3, over the left cortex. So we then give them a cursor that moves up and down, and we say, okay, the up and down movement of the cursor is going to be controlled by the amplitude of the mu rhythm in this location. Try to use various kinds of imagery to see if you can hit a top target or hit a bottom target. And people, by trial and error, generally learn, and this is something that develops pretty rapidly over the sessions. They're like 8 3-minute runs in a session, and over 2 or 3 sessions, the majority of people acquire some control.
If I were paralyzed, I might think in terms of a if I could lift my right hand.
Yes, right. You might think about moving, about relaxing, about floating. People came up with all sorts of different things. But as they get better, and especially as they go on to 2 dimensions, that imagery kind of drops away and it becomes more automatic. Yes?
How many electrodes and how much of the motor area you must control to get the signal?
Well, we normally record. Oh, the question is how many electrodes we use to control this. Generally, we always generally record from 64 electrodes, but that's so we can see everything that's going on. Generally, for say 1-dimensional control, we're using 1 location that we've chosen, 1 frequency band. For 2 or 3-dimensional control, we might be using 2 or 3 locations and various combinations. That's going to vary from person to person, but it's really a very small number that actually does this online.
Question. And is the reason that you use a limited number of regions because of computational limitations, or is it because of some sort of human-related thing?
Well, we try to go 1 dimension at a time. So we're looking for something to control 1 dimension. Generally, sensory motor cortex is the only area we've ever had real success with. And clearly, we've tried other areas, and basically you don't get the training effect that you get with sensory motor cortex. So we confine ourselves to that.
But what if you were using multiple areas to control 1-dimensional or
right. Would you be better?
two-dimensional?
Potentially.
Like, because the idea of your brain being a Bayesian
Yes.
network and all of
Yes.
the neurons contributing, and that may take down on the shake.
That's potentially, but I think from what we know, it would be a relatively incremental improvement that we would get by that. So, going back, okay, so here is as we understand, a very simplified diagram of the nervous system. It's been evolved over millions of years. It's developed through all our lives for 1 purpose. You have all these areas that are interacting. They're all plastic, and all they're doing, as far as we know, is controlling motor neurons that control muscles that produce actions. So they're optimized for this. They've been optimized through evolution, and they're optimized through development to do this. And they do it very well and very reliably. A BCI changes that. A BCI asks all these areas to now produce very accurate control of cortical neurons or rhythms, sensory motor rhythms, some measures of brain function from the cortex, and that becomes the output. That becomes the control. We're taking this system that's configured to do that, and we're asking it to do that. We're asking it to change itself to do that. And the answer is that, yeah, it can do it, but it can't do it all that well, at least in our hands up to the present. And that's where the Grand Canyon problem, I think, comes from. So people are looking at this in various ways. There are looking at multiple areas, as you mentioned, is one way that can get some improvement. Providing better somatosensory feedback rather than the very crude visual feedback we normally provide can help. But I think there's some very fundamental problems as to how the nervous system adapts that we really have to address, and there's some important principles that I don't think we know as yet to really make this work as reliably as this. So at this point, for practical purposes, BCIs are the ones that can actually help people with communication control are relatively simple, like the P300 based BCIs.
Question here.
Yes.
You know, I've seen a number of years ago a demonstration of people who have lost the ability to talk, and rather than trying to do a BCI, have a collar on their neck, since even though the vocal cords perhaps have been destroyed, you can pick up the electrical signals within the neck. So, and yet your research seems to focus only on the brain versus picking the same signal up from the fingers or the hands or other parts and make it a multifunctional thing, which gives you a whole nother realm of potential smoothness.
I mean, you're very right, and there are lots of potential ways to help someone who's paralyzed. A BCI, by definition, is taking signals from the CNS, but there are many non-CNS ways. We were talking earlier about targeted reinnervation, in which you use reimplant peripheral nerves that have been cut off in an amputation. You implant them in the chest muscles, and you basically use the muscles of the chest as amplifiers. And so you can record EMG, and people can learn to control a robotic arm, a prosthetic arm, very accurately. That's not a BCI, but it's great. And you're really using it in a normal fashion. So yes, there are many other ways, and that's part of the reason why the Grand Canyon problem has to be solved before these systems ever come out of the lab, because there are lots of other ways to help people who are paralyzed.
Yeah, I was referring more on your last diagram. It shows that you're trying to replicate the output of the motor neurons,
Right.
and there still may be some small output from them
Yes.
that you can
yes.
combine with the BCI to a great advantage.
Yes, there are combination hybrid BCIs are a target of research. Yes, but you're right. So, but there's another area of BCI applications that I mentioned, improve, which doesn't have the same high bar. You don't need completely reliability, consistency. If you can show that a BCI improves normal rehabilitation, that it gives a better final result, that it's effective, it doesn't have to be perfect. It can be something that could be used clinically, and if it worked, say, for people with strokes, it would have a huge impact. So that's an area that people over the last 10 years have started looking at. This is from a paper that Janice Daly and I wrote now, well, about 9 years ago, looking at the ways in which, and I'm not going to go through this in detail, a BCI might improve rehabilitation. One way is to directly train signal features such as sensory motor rhythms so that they're more normal, to lead to more normal movement. Another is to use brain signals in a movement assist device to make more normal feedback. And both of these could be conceived as leading to enhancing recovery. There have been a number of efforts now, they're growing over this, to do this. The sensory motor rhythms are one area that's received particular attention. As I said, they reflect the brain activity underlying movement. And this shows, for example, the amplitude of a particular sensory motor rhythm goes down as you prepare for movement. This is when the movement's occurring, someone's preparing, the amplitude of this rhythm goes down. For example, this would be before right hand movement over the left sensory motor cortex. The sensory motor rhythm is getting smaller. And we showed that if people decrease the sensory motor rhythm prior to movement versus increase it, they move faster. The movement time is less when they have lower rhythm before movement. The movement is more accurate when they have lower sensory motor rhythm before movement. So this actually affects performance. Modifying them thus might improve impaired movement. And this, I think, is the first actual controlled study that appears to demonstrate. This is from Donatella Mattia's group in Rome, came out several years ago. And what they did was they were looking at people who had strokes, who had impaired movement of their hands, and they were putting them through two motor imagery training protocols. In the control condition, the person just had trials in which he's being told to imagine opening his hand, whatever that means to them. So he's practicing imagining opening his hand. In the other trials, and you can see EEG being recorded from the surface from his scalp, the computer is looking at a sensory motor rhythm, and he's being provided feedback as to how much desynchronization he's producing when he's imagining moving his hand. So he's improving the desynchronization. He's learning how to improve desynchronization to move a cursor on a screen. And as a result, when you compare at the end the control population to the BCI population, the control population had increased desynchronization during movement over the contralateral cortex. This is in the alpha band, that's 8 to 12 hertz. The beta, which is like 18 to 26 hertz. And this would be for a right-hand lesion. There's increased desynchronization. There's increased connectivity in the BCI group between locations on the scalp. And most important, there's increased improvement. These are different measures. This is the Fugl-Meyer scale, the Medical Research Council scale of arm function, and the NIH scale. There was considerable improvement, and this is in percent of their deficit, the percent of difference from perfect performance. Perhaps a more meaningful way of saying this is that in the BCI group, about 80% of them had what was considered a functionally significant improvement in motor function, the Fugl-Meyer scale, at least 7 points. And in the control group, it was only 20%. So there's evidence here that the BCI actually enhanced rehabilitation. So this is an area that's getting increasing attention, and I think probably in the foreseeable future will be the BCI area that has the most clinical impact. So another kind of adaptive neurotechnology is the use of electrocorticography. These are signals recorded from the surface of the brain to localize cortical function before surgery. So when you do electrophysiological imaging of the brain, you can record EEG from the scalp. This is pretty much the scale. You see the scalp, the soft tissue, the skull, the dura. Here's the cortex with neurons in it. You can record EEG. You can record ECoG from the surface. You can record with microelectrodes, spikes, and local field potentials within. So I'll be talking about ECoG, cortical surface recording. Imaging brain processes, there are 3 kinds of requirements. You need spatial resolution, you need temporal resolution, and you need coverage. So EEG has good coverage and good temporal resolution, but it has bad spatial resolution. fMRI has good spatial resolution and has good coverage, but it has really lousy temporal resolution. That's functional magnetic resonance imaging. Intracortical recording, say with microelectrodes, has great spatial and temporal resolution, but it has lousy coverage. You're not going to put microelectrodes in the entire brain. So ECoG really has satisfactory performance in all these areas: spatial resolution, temporal resolution, and coverage. So this has first been applied to ECoG, in ECoG-based functional mapping prior to surgery. In epilepsy surgery, you often need functional mapping, so you know what to take out, for example, or what you can take out, what to take out, and what you shouldn't be taking out. And normally what happens is an array of recording electrodes are put over the relevant area of cortex for a few days or a week prior to surgery to enable the surgeon to localize where important functions are. The traditional method that's been used for close to a century now, actually, is to stimulate at a particular electrode. Have someone do something, have them talk, have them move a hand, and then stimulate at that area, and just stimulate higher and higher until you can interrupt the function. Now this takes a long time. There are a lot of electrodes, and it's dangerous. You can evoke seizures with this. But it's essential to be able to localize function. So ECoG-based passive mapping is a promising alternative. This is a new method, a new mapping method called Siegfried, developed primarily Gerv Schalk and Peter Brunner in our group. This is a complicated slide, but I'll show you. This is a person who has an ECoG array implanted. You can see she's sitting. She's going to be doing a variety of tasks. This is the side of her brain from MRI. This is the array of electrodes that's placed really right here on her brain. Oh, okay. This is ECoG recording you can see here. And these are the instructions that she's getting. So at the beginning, she's just relaxing. And what happens here is she's going to do different things, like solving Rubik's Cube, and the computer is going to determine which locations have different activity when she's doing this than the activity they had when she was relaxing. And you'll see this happens very quickly. So this is all in real time. Now she's solving Rubik's Cube, and you can see the areas that are involved, gradually isolated. This is all in real time. You can see on her brain. These are the areas that are participating in that, and it's done. And now she's relaxing again. And then she'll be sticking her tongue in and out in the second part. And so it's now finding the locations. These are the locations, and this, the size of the circle is an index of how the level of probability that this is different from what she was doing when she was relaxing. And you'll notice during relaxation, these guys continue to change because the system is getting increased information about what relaxation looks like. Now she's going to be making kissing movements. Here. And the last one is very interesting because it'll be very different. The last one is auditory input.
How long does it take for relaxation?
Pardon?
Mr. Jones had
Wait,
to
wait.
go to New York. He decided to take a train. His wife drove him to the station, but on the way they had a flat tire. However, they arrived at the station just in time for him to catch the train.
So you see the activity is here where you imagine it over primary and secondary auditory cortex. So there in a couple of minutes, you've done a analysis, functional localization that would have taken a very long time with stimulation. Yes, you had a question?
The time it takes to relax.
Pardon?
The time it takes to relax.
Yes.
In every step, as you're doing the 4 steps.
There's a pause in between,
Yes,
in which is a few seconds, as you saw. It's about as long as you saw. It is as long as you saw.
The whole thing in about a minute or so.
So this, well, however long this took, 2 or 3 minutes, that's the whole thing for those particular functions. Yes?
But even during the relax phase, it didn't seem like some of the areas went to zero as before.
No, it was still updating its information.
Right, but some looked like fairly large, so I would
Yes.
have thought the kissing might wipe out the sticking out the tongue, but...
No, because that's a separate analysis. Basically, it has this base of relaxation data, which is continually getting bigger. And it's ongoing as there's another period of relaxation. It's now got 2 or 3 periods, which it's comparing to the Rubik's Cube data, and which compares to each one of the, it's a separate comparison. So, yes. Well, this I think was just a demonstration. Yeah. So, yes.
With the stimulation protocols, you typically map speech production and speech understanding.
Right.
Do you guys have the ability to do that yet? Because obviously auditory input is different than
Right. They've looked at listening to music. I believe they've looked at actual vocalization during or sort of internal vocalization. They've looked at a variety of other things with this. Not at, I'm not sure. What, yeah.
Use the microphone.
Yes.
So
Right.
with typical ECoG, you
Right.
and especially with the passive mapping, you either map language production or language understanding. This is a fabulous demonstration, but it wasn't clear that we're able to figure that out yet
Yeah.
from this passive
I
mapping.
believe they've done more, and you really
Okay.
have to ask
Because that's the
Gerb or
critical
Peter about
thing
it,
that
but
you
yeah.
do for.
Yeah. Pardon? Yes. Yeah, he's actually, we're working with.
Yeah,
with
but
him on
Bob
that,
Knight
too.
doesn't approach it like this, which is why I was asking.
Well, actually, he and Gerb were working together some, so I think maybe he's using this now. I should also mention as a disclosure that this technology is being marketed for neurosurgical procedures by Cortec, and I and others who are involved in it receive a modest, and I emphasize very modest, licensing fees for it. So that is for clinical purposes. At the same time, this same kind of technology is being used in basic science to actually trace on a single trial basis the movement of an action through the nervous system. So this is an example of a reaction time in which there's a visual stimulus and the person then makes a hand movement in response. You can use it on a single trial basis to track this process going through cortex. So it starts here, and then you see activity appearing at other locations, and finally arrives in the sensory motor cortex where that produces the action. And you can explain reaction time. You can explain why this trial, the person responded faster than that trial by looking at, on single trial basis, at the rhythms at each location. So they're using this now for basic science purposes too. And they're also going on to look at this for functional imaging under anesthesia. They're just at the beginning of that. So I'm going on to now to the final area, which is reflex conditioning for rehabilitation after spinal cord injury, and this introduces the idea of targeted neuroplasticity. So in traditional rehabilitation, starts from the assumption that the central nervous system is largely hardwired with little capacity for plasticity. It tries to maximize the remaining capacities and to induce whatever minimal plasticity is possible. There are really two major strategies. One is to practice impaired behaviors, such as locomotor training, or giving drugs to reduce dysfunction, drugs to reduce spasticity, for example. There's often limited efficacy, and important behaviors are not fully restored. Significant side effects may occur, particularly with the drugs. So we now know that plasticity is ubiquitous and continuous in the CNS. There are many different kinds. There's neurogenesis, gene activation, neuronal properties change, there's long-term potentiation, long-term depression. You have glial plasticity, dendritic plasticity, hormonal plasticity. You have sprouting, you have vascular changes. All of these kinds of plasticity conspire to produce behaviors. So this activity-dependent plasticity, which is occurring continually throughout the nervous system, has many mechanisms and probably trillions of sites. And the time scales go from milliseconds to years. Thus, when function is impaired, there are many options for how it might be repaired. However, there's no direct overall guidance. There's no overall oversight that's saying, oh, this person is not walking properly because of a spinal cord injury, so we need to create plasticity here, 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 and here, and now he'll walk better. There's no overall oversight that's going to do that. The drivers of plasticity at each site are local. Most sites are far from behavior. A particular synaptic connection doesn't care whether it's increased or decreased activity has any good or bad impact on the behavior of the person. All it knows are the drivers that are in its immediate vicinity that control the factors that make it change. So there's no certainty that the optimal pattern of plasticity will occur. Traditional rehab is not specific. It doesn't target specific sites. It's seldom fully effective. New methods are needed. So this is the idea of targeted neuroplasticity. So here you have a nervous system, that's the white, with a bunch of sites that could change. Those are the deltas. It's acquired one behavior that's involved changes at a number of sites. Trauma or disease occurs. These sites become abnormal and the behavior is abnormal. If you can go in with a neurotechnology protocol and force an adaptive and a beneficial change at one key location, you can set up a protocol that will force change at that critical location. You can lead to wider change that may largely restore the behavior or restore it more effectively than traditional rehab did. So the way we've done that initially is modifying the pathway of the spinal stretch reflex. This is the knee-jerk reflex. And when you stretch the muscle, you produce input that comes in on the sensory axon, synapses largely monosynaptically on the motor neuron, goes back and leads to the contraction of the muscle. That's called the spinal stretch reflex. You can also do this electrically by giving a weak stimulation directly to the nerve, in which case you stimulate some of the largest efferent fibers, which go out to the muscle and produce what's called an M response or an M wave. You also produce sensory input, largest afferents, which go in, excite the motor neuron, and produce what's called the H reflex. It's called H because it was described by Hoffman now almost 100 years ago, I believe 1918. So this is We use the H reflex as opposed to the stretch reflex, which we used initially, simply for that it's experimentally much more convenient. So this is a totally spinal pathway, but it's subject to descending control from the brain, both on the motor neuron and on the synaptic connections. So the brain can affect the size of this response. Here you can see this reflex arc. Here's an incoming sensory afferent fiber, synapses at various places. This is a labeled soleus motor neuron. You can see synapses that are synaptic connections probably on the dendrites and proximal dendrites and cell body of this motor neuron. What we and now others have shown is that monkeys, rats, mice, and people can gradually change H reflex size when they're rewarded for doing so. And this is a story that really goes back quite a ways now. It began as basic science to look at plasticity in the nervous system in the monkey stretch reflex. It's an example of how basic science eventually yields clinically valuable applications. Through a whole long series of mechanistic studies in rats and in mice as well, and more recently, rats with spinal cord injuries, and finally in people with spinal cord injuries. So this is the development over now almost 40 years, which what began really as basic science. And the basic principle here is operant conditioning, is a situation in which a subject is given a specific stimulus or placed in a specific situation, and reinforcement or reward occurs when a specific response occurs. After repeated exposures to this experience, the required response occurs more frequently and thereby increases the number of rewards. So what we're really conditioning here is the descending control. We're getting the people or the animals to change this descending control to change the size of the reflex. That's what's being conditioned. And this is the situation in a rat. They're recording electrodes in the soleus muscle, a stimulating cuff on the posterior tibial nerve. The rat, all this goes through a flexible tether to an amplifier and stimulator. The rats are online 24 hours a day for months. They can move freely about their cages. We're simply monitoring their EMG. Whenever the EMG is in a certain range in the soleus muscle, we elicit an M wave and an H reflex. In the control condition, we simply measure them. In the training condition, we reward the animal with a food pellet if the reflex is above if we're training him up, or below if we're training him down a certain size. So this is the basic phenomenon you see in many rats trained up over 50 days, other rats trained down. This is the control period here. This is a rat trained up. You see the M wave and the background EMG haven't changed. The H reflex is bigger after up conditioning. In this rat, it's smaller after down conditioning. The same phenomenon has been demonstrated in people by Eiko Thompson, who began this work as a postdoc in our lab. It's a very similar setup. The people are not implanted. They're not online 24 hours a day. They perform 3 sessions, 3-hour-long sessions 3 times a week. But the phenomenon appears very similar. They receive feedback indicating whether the reflex has been the correct size. And this shows the phenomenon in people. This is over sessions distributed over 8 weeks. People trained up, people trained down. The persistence of the change months later. This is an increased reflex, the dashed line. This is in a person trained down. So we've spent a lot of time looking at the underlying plasticity. All the pink areas are sites of definite or probable plasticity associated with this. There are multiple changes at the spinal cord level. You can see, for example, GABAergic terminals with down conditioning become much more prominent on motor. This is a labeled soleus motor neuron in a normal rat. Untrained rat, you can find a few GABAergic terminals. In an animal that's been trained down, you find many more GABAergic terminals. These are probably terminals that previously weren't expressing GABA. Also, there are changes in the firing threshold of the motor neuron with down conditioning. The firing threshold becomes more positive, so an EPSP, which previously would have caused the neuron to fire, no longer does. So there are multiple changes at the spinal cord level. There are also changes in sensory motor cortex probably and in cerebellum as well. Our current understanding is that this really is a hierarchy of plasticity in which the inferior olive guides plasticity in the cerebellum that guides and maintains plasticity in sensory motor cortex, that guides and maintains the plasticity in the spinal cord that directly underlies the change in the reflex. So how does this relate to rehabilitation? This is a rat, this is a normal rat spinal cord. In the rat, the corticospinal tract, which is the only pathway that's essential for this phenomenon, is conveniently at the base of the dorsal columns. So we can go in and destroy the entire lateral column in a rat, and it's still capable of conditioning. This is a normal rat walking along. These are the soleus bursts on the right and left side. Here you see as the rat walks along, this is the onset, the solid is the onset of right stance, and the open is the onset of left stance. So this, and the dashed line is halfway between the right stance. So this animal is walking along symmetrically. This is a rat with a right lateral column injury. The right soleus burst is not long enough. It's not sustained. And as a result, the animal's limping. So it's walking like this because right stance isn't adequately maintained. So can we go in and make the burst stronger by conditioning the reflex up on the right side? And you can see before and after, we can make the burst bigger after a spinal cord injury on the right side. And as a result, this is the animal before, and this is the animal after. It's now walking symmetrically. We haven't made the spinal cord better, we haven't repaired it, but we made this function better. So after conditioning ends, the beneficial effects, and just this is the relevant data here, animals trained up, walking gets better. After we stop conditioning, the reflex continues to increase and walking continues to improve. We've triggered a process that continues after we stop because it's beneficial. What about in people? Now in people, the problem is different from that in the rats. They don't have weak stance, they have spasticity that impairs their locomotion. So in people, the logical strategy is to decrease the soleus H reflex. And this is ECoG's initial study in people with incomplete spinal cord injuries. They learned to make the reflex smaller. This is an indication of the result. This is a person walking along before. There's clonic activity. This is the burst, the locomotor burst. There's abnormal clonic activity occurring here during the stance period, during the swing period. So the foot is jerking up and down. It impairs movement. Afterward, it's gone, largely gone. The clonus is gone after the down conditioning. You can see here, this is a normal subject walking along. This is soleus activity, stance and swing. This is the opposite muscle. This is the front of the calf muscle that lifts the toe. It has high activity at the onset of stance, so it keeps the toe up so you don't stumble over your toe. And this is the excitability of the reflex, of the soleus reflex. It's low during the swing period. Here's a subject with spinal cord injury before. There's clonic activity here. This is the foot jerking up and down. There's not tibialis activity to prevent toe drop. So this person has foot drop and clonic activity, and the H reflex is much too big during the swing period. This is after being trained down. You have a better looking soleus burst. It's not perfect, but it's better looking. You now have activity here that keeps the toe up, and you have decreased H reflex excitability. Improved soleus and tibialis anterior EMG after conditioning. The targeted plasticity triggers wider plasticity. Again, we've just trained the H reflex in the soleus muscle in one leg. It improves, that's the dashed, soleus activity during locomotion in that leg. In the other leg, it makes tibialis anterior activity more normal. It also produces better locomotor modification in the vastus lateralis and biceps femoris muscles in both legs. So we've caused a much wider improvement. Walking speed and symmetry improve. This is the decrease in the reflex. This is the improvement in walking. This is the improvement in symmetry toward 1. So right-left time is the same as left-right time. And you can see here in the subject the decrease in limping. This is the onset here is too late. After down conditioning, it's occurring on time. And these are comments, spontaneous comments people made over the course of conditioning. These are the people who decreased the reflex. These are people who did not decrease it, either because they were straight controls or because they weren't able to decrease it. And this is the point at which they said things such as, I'm walking faster, I'm walking farther, I have improved balance, etc. These comments were almost exclusively by the people who decreased the H reflex, and they didn't begin till sessions 12 or 18, when the reflex had reduced by a considerable amount. So this is an indication that gait improvement was apparent to the subjects.
How is it? How does the training make changes in your movement, in your control, in your mobility? The results to me honestly have been incredible. Not coming and not expecting anything, but just to hopefully help out the study. I really surprised myself. The first thing I noticed was an increase in my speed. You know, it's still not of a typical healthy person, but I'm definitely able to carry my leg through more consistently, smoother. I used to really try to rely on a hip flexor that I don't have to try to walk, and I've learned to recruit other muscles that I didn't even know I was capable of using to walk. My spasticity has significantly decreased. The clonus, the shaking spasticity has. So just that loosening of the muscle has definitely had a positive effect on me. I don't get fatigued as easily when I am walking. I actually, at times, felt like my gait was almost normal. And for me to say that is a very, very bold statement. But I completely mean it.
So that's targeted plasticity of the H reflex arc. I'm just going to show at the end a little bit of, Eiko is now using the same protocol to strengthen corticospinal connections. This is at her lab at MUSC. We continue to work closely with her. The sessions are very similar, but you can see transcranial magnetic stimulation, activating the corticospinal pathway, looking at the size of the response in the tibialis anterior muscle. This is the muscle that lifts the toe to prevent foot drop. This is to strengthen that muscle, the participation of that muscle during locomotion. If this training protocol is the same, you can see that you can actually train it in people with incomplete spinal cord injury. It improves the maximum voluntary contraction. Here you can see the increase in the size of the response, but most important, it improves walking so that you see a much better soleus burst afterwards. You see much better tibialis anterior activity. Before there was foot drop. Afterwards, you can see tibialis anterior activity to keep the foot up. And this persists 6 months later without any continued conditioning. This is in people, she's also begun to study people with multiple sclerosis. Again, they're able to increase the size of the burst. You can see the increase in the size of the motor evoked potential elicited by TMS. There's an associated increase in maximum voluntary contraction, and there is improvement in tibialis anterior activity during locomotion, with a consequent improvement in the speed of locomotion. So, in summary, recent scientific and technical advances enable a new class of technologies that interact directly with the CNS, that take advantage of the plasticity that we now appreciate is ubiquitous in the central nervous system, to take advantage of it, to guide it, to use it to improve function. Brain-computer interfaces to restore communication and control are one potential use of this that's received a lot of attention. At this point, the simplest versions of BCIs for communication and control are really all that's ready to be taken out of the laboratory. The more complex methods require some fundamental problems to be successfully solved. But BCIs to enhance rehabilitation may be in use much sooner and could have a much bigger, much wider impact. Because in that case, they only have to be effective. They don't have to be perfect. And we're beginning to get indication that that's possible, that they can be effective. BCI, the adaptive neurotechnologies can be used for safe, rapid cortical mapping prior to surgery. The development of the same kinds of techniques can be used to dissect cortical function, the timing of cortical function in ways that were previously not possible. And finally, protocols that can target beneficial plasticity to critical sites can trigger wider beneficial plasticity to improve complex behaviors such as locomotion. So it's clear that if you can make a critical change, you can initiate much wider beneficial changes. I've talked about operant conditioning protocols. There are also stimulation protocols that can target plasticity as well. So there have been a lot of people involved in this over the years. I've talked particularly about Shunyan Chen's work in the rats with conditioning, Dennis McFarlane, the BCI work, Gerv Schalk, the work of cortical localization, Teresa Vaughn with our clinical applications in BCIs, and collaboration now with Iko Thompson in South Carolina looking at clinical applications of reflex conditioning. Jonathan Karp has also been very much involved in the basic science in our rat studies. So we've had support, as I said before, for many years from NIH, NIBIB, our P41 Center as well for many years from NINDS and from NCMRR of NICHD. And more recently from the VA, the Army Research Office and DARPA, and a number of private foundations. I should announce that we have an NIH-supported short course in adaptive neurotechnologies. This is a 3-week course in the summer. The first edition was last year. It's going to be this year in mid-July. We already have the 24 students for that, and it will be done. It will be advertising it for next year as well. So thank you, and I'm happy to answer questions.
Very excited. So we have some questions. If you go to the microphone, this one, maybe. Well, let me slip in a quick question. In the examples you showed of the operant conditioning, with the are there changes in the fMRI imaging that indicate changes in plasticity?
No, basically, we know that there are changes in the brain because, I mean, I'll just give you one fragment of what we know. If you eliminate cerebellar output to the brain, you don't get any down conditioning. However, if you down condition and then eliminate the cerebellar output, most of the down conditioning remains. However, if you wait 40 days, it disappears. I mean, we didn't believe this the first few times we saw it, but it happens at 40 days. You destroy the cerebellar output, and 40 days later, the conditioning disappears, and you wind up with a reflex larger than it was to begin with. Almost the same thing happens if you destroy the inferior olive. The form is a little different, but it's clear that the cerebellum is maintaining plasticity, probably in sensory motor cortex, that can survive for about 40 days on its own, and then it disintegrates. This is something we don't know the nature of this. We just know that it's there, and we don't have any MRI studies as yet of this.
Does it work the other way around? That cerebellar damage alters the adaptive?
It's the ability to change the reflex, but cerebellar damage itself does not affect the reflex in an untrained animal, which is very convenient for us. But
you gave an example of the effect of transcranial magnetic stimulation on the spinal cord injury. So at what level of the injury was there in the spinal cord? With the MRI, probably you could see some of the...
it was between the, It was an incomplete spinal cord injury that was between the brain and the site of the TA in the
So, I
spinal
mean,
cord.
how much damage was there?
Oh, and that one I don't know, but it was not a complete injury.
So is there a threshold when you select the patient that beyond certain
well,
damage you will not
yes,
accept the patient?
at this point, people will have to be able to walk in some fashion. Some of them are extremely slow and they need walkers or canes, but they can walk in some fashion. And in fact, as I didn't mention in our initial study, there were people who began in crutches and afterwards were using a cane. Another person was using walkers and afterwards was able to use a cane. But these are people who have impaired locomotion. It can be very impaired.
Yeah, John, have you thought about conditioning this upper extremity stretch reflex to improve upper extremity
ECoG
function?
just started, and we're starting to do that too, looking at the flexor carpi radialis. We're just setting up to do that, and it looks like that's possible. We're setting up our technology now. So.
You seemingly had a lot of amplitude time responses to the outputs of the electrodes. Do you look at FFTs or wavelet transforms of those as well?
For in the BCIs, or?
Yeah.
We look at basically FFTs, or autoregressive frequency analysis. We have not looked at more complex analyses, mainly because we haven't, I mean, a lot of people looked at very complex EEG analyses. We've never seen anything that works better than the simplest methods. There probably is, and the other thing is that you have to be able to adapt. You have to be continually adapting to the person from one day to the next, to the learning, et cetera. And if you're looking at something very simple, it's easier to adapt. So Dennis and I have been in complete agreement. We don't a lot we don't agree on, but in that we're in complete agreement that we keep things as simple as possible. So.
This is very interesting. Now I believe in this device I came across in the web that claims it can improve gait of injured person through the stimulation delivered through the tongue to people. But my question is about reward. Do you find there are different efficiency with delivering reward in different ways? Through visual stimulation or food, or maybe directly through the nerves somehow? Yeah,
we've done a little at the very beginning in the rats. We thought we wouldn't be able to give them a small enough food pellet to get enough trials, so we were using medium forebrain bundle stimulation as the reward, and that worked. But it seemed to work. It turned out food worked just as well. We didn't really see any difference between the two. We haven't looked a lot at the nature of the reward. Clearly, we probably should. It could be better or worse. In the monkeys initially, we tried to give a graded reward. We were giving them a liquid squirt, which went in their mouths as a reward, and we figured, well, if they do extra well, we'll give them 2 squirts. But what would happen is the monkey would open its mouth, get hit by 1 squirt, close his mouth, and the other one would hit him. So it was basically counterproductive. So we just stuck to a very digital, yes, no reward. But it's an area that needs investigation.
Okay. Well, listen, Jonathan, thank you very, very much. That was very exciting, and we appreciate it.
Thank you.